Now we're going to get into the nuts and bolts of the basis of inheritance, and that centers around DNA replication. You might have studied a little bit of this in the past, but we're going to drill down into these phrases and so forth in this chapter. But we're going to do this in layers. We're going to do this incrementally. So let's see how this goes. The first thing I will say is that there are literally thousands and thousands of wonderful images and resources online for DNA replication. So if for any reason this lecture isn't working for you, please don't hesitate to talk to me and we can find you some other resources that may make more sense for your learning style or so forth. At the end of the day, we still have to learn about the different pieces, like this picture right here, or even this picture right here. This is from our book, by the way, and this is also, we're going to be in this region here of detail. So if you have any question about that, please let me know, and I'll try to steer you in the right direction. Given that we have to replicate our DNA literally hundreds of millions or billions of times every day, our entire lives, trillions and trillions of times over the span of our lives, it's very, very important that we have a mechanism and we have a system that will not only replicate DNA in a faithful way, but also repair it should it gets damaged for any reason whatsoever. And so when Watson and Crick developed their double helix model, they immediately saw a potential mechanism for copying DNA in a way that would be relatively low error prone. And so they suggested this in their original paper, just implied it. They didn't get into the nitty gritty, but they did imply it. And it didn't take all that long to establish that in fact their hunch was correct in the basic mechanism for copying the DNA. And so we're going to talk about that basic mechanism, and then we're going to get into the nuts and bolts later on in the end of the lecture. And the genius and elegance of this double-stranded molecule is that you can use each strand as a template to synthesize DNA, another strand. So you can use the old strand as a template, synthesizing new DNA using the old strands, and thus you end up with two identical copies in a relatively short order. You don't have to go through a lot of gyrations to get there. So when we replicate DNA, one of the things we have to do is we have to unwind the old DNA and synthesize the DNA on using that older DNA as the source of information for making the new one. Here we have a parent cell with its parent chromosomes over here on the left. What we do is they call this melting, by the way. When you melt the DNA strand, you are separating the two strands. You're breaking the hydrogen bonds, and so you're separating the two strands, and you're synthesizing the new DNA using the base pairing rules on the old strands. So you use the old strands to say, hey, I need a G, or I have a G here, I need a C here. I have an A here, I need a T here, and so forth and so forth. This is known as semi-conservative. This is semi-conservative DNA synthesis. What semi-conservative means is that you're going to have in each daughter cell, each daughter DNA molecule, you'll have one parental strand shown here in dark blue. So we start with two parental strands here. One parental strand is a template, one parental strand is a template. And then if we do one more round, what you'll see is that original parental template is now only in one of these. So you can see this. And that's the basis for how we determined whether it was semi-conservative or fully conservative or completely what we call dispersive. So the competing models were that you used the old DNA, but you made an entirely new strand. Or dispersive, where you had little pieces of old and little pieces of new mixed together. So over here on the left is a conservative model where you end up with an entirely new strand, double strand of DNA that does not have any of the original parental. And so after a second round, you will have only one old and three new. Whereas with semi-conservative, you would see that you would split that, and then it would split again. Whereas dispersive, you have little fragments here and there of old and new. And we're going to show you the experiment that proved that it was in fact semi-conservative. And again, data interpretation is one of the skills that you're going to want to have when it comes to answering questions on the exam. Here's just another picture showing conservative versus semi-conservative versus dispersive, and that's from your book. Experiment that really sealed the deal when it came to semi-conservative were run by Messelson and Stahl. Very, very clever. It's non-radioactive, but it does use an understanding of atomic weight. That is to say, they grew up bacteria in heavy nitrogen, nitrogen 15. So they grew up, and so all of the nitrogenous bases in the DNA of these E. coli were heavy, were seeded with heavy nitrogen. And I mean by heavies because they will have a higher atomic mass and they will actually have more mass than if we were to grow up the bacteria in 14. So they would let the bacteria grow in nitrogen 15, and then they switched the broth to nitrogen 14, and then isolated the DNA after a certain number of divisions. So they grew them up in heavy nitrogen, nitrogen 15. They transferred those bacteria to nitrogen 14, and then they let them grow for one cell division. One cell division. So they let them do one cell division, and then they isolated the DNA and centrifuged it. Okay? They isolated the DNA and centrifuged it. So after the first replication round, what they saw was only one band. If it had been completely conservative, you would have still had your heavy nitrogen DNA strand, and you would have had a new lightweight DNA strand made with nitrogen 14, because they would have been using the nitrogen 14 to make the new strand. But they didn't see two bands. They only saw one band. So that certainly rules out fully conservative, but it does not rule out dispersive. Remember, dispersive is where you have little bits of old and new mixed together. So you would have seen a band that would have had a similar atomic weight, even if it was dispersive. So we rule out one model. We rule out fully conservative, because if it had been fully conservative, you would have had a lightweight band way up here at the top, and you would have had a heavy band lower at the bottom. Okay? But after the second round, it rules out dispersive. Because if it had been dispersive, you would have seen one band, because again, you're mixing lights with heavies. Okay? But what we saw in the experiment was we actually saw a light band and then an intermediate band. Heavy band would have been down here. You can predict that, by the way. But you see the original intermediate band there, and you see a new light band. If we go back and look at the patterns, you'll see why it would be completely light, because those ones, now you're using one of the lighter bands as a template. Again, if it was dispersive, you would have seen an intermediate band, only one. So let's look at this side by side. First replication, if it had been conservative, you would have had a heavy and a light band. Okay, we didn't see that. You would have had a light band from the new synthesized DNA and a heavy one. Didn't see that. We didn't see a heavy band at all. All we saw was this intermediate band. Heavy band's way down low, light band's way up high, intermediate. Dispersive would also be intermediate, but it's the second round of replication that seals the deal, because we would have had, again, a heavy and a bunch of light. What we see now is that intermediate again, not the heavy, and light. What we don't see is that intermediate. Now it's a little bit lighter. Why? Because you're synthesizing more DNA. There's less overall old DNA relative to the new, and so that band would gradually shift up. You'd see a little more lightweight band there, because as we go, the old DNA becomes more and more rarefied in that dispersive model. Didn't see that. We saw two bands. We saw that intermediate band, half heavy, half light, and we saw the all light. And so that sealed the deal on semi-conservative. And just for example here, this is actually some DNA that I spun. This is my own DNA prep here. You have it labeled here. And you can see the band here of DNA after we were purifying it. So you can see it right there. It's a really, really fun thing to do. It's like doing art. A lot of little teeny tiny pieces of DNA up here that we don't want. We want to get rid of that stuff, and so we get this nice enriched band at the molecular weight that we're looking for. And then what we do is just stick in a syringe on the side and draw that out. So that's the same experiment, only they did it with their bacteria, and I was doing it to purify some DNA that I had prepped up in the bacteria. So now that we know that DNA is semi-conservative, you have this parental DNA that's being unwound, and you're synthesizing new DNA on top using the old one as a template. We now want to talk about the mechanism of replication, but we're going to see that we're going to have some technical issues. Why? Because these strands are anti-parallel. Remember that one strand is going in the opposite direction of the other. And so what we'll find out is that we can only synthesize DNA 5' to 3'. We can only add nucleotides 5' to 3'. Well, that introduces a little bit of a technical issue when we're unzipping the DNA, as it were. Because one way we can just synthesize it straight away. The other one we're going to be going backwards. So we've got to figure out how to solve that problem. And that's what we're going to be talking about. And that's really where the complexity sort of becomes, is in what we call the lagging strand. Leading strand just synthesizes as you unwind. Lagging strand is going to be a little trickier. Really cool is that in bacteria, which actually have circular chromosomes, you can actually witness this firsthand. You can actually see the DNA unzipping this way and the DNA unzipping this way. Remember that we can only synthesize DNA from the 5 to 3' direction, which means we're reading 3 to 5. We're reading 3 to 5, synthesizing 5 to 3. So if this is anti-parallel, over here that's no problem. We're reading 3 to 5, synthesizing 5 to 3, but what about the other direction? And so we have what's known as a leading strand that is reading into what we call the replication fork. And we have what's called a lagging strand, which is working away from the replication fork. Over on this side, into the replication fork, that's a leading strand. And away from that replication fork, that's a lagging strand. So we need to understand leading and lagging strand. They're both synthesizing DNA, so we're synthesizing both strands simultaneously in this direction. Both strands and simultaneously in this direction, but one strand is always going to be leading and one strand is always going to be lagging. So where do we get started in the DNA replication? How do we get this going? We can't just melt DNA any old place. That's true in the most simple organisms and in the most complex organisms. What we have found over time is that there are special parts of a chromosome which seem to be more amenable to melting, more amenable to strand separation than others. In the case of bacteria, it is a specific spot. You have one exact spot that seems to have this special DNA sequence. In eukaryotes, you tend to have many of them. But in any case, it's what we call a replication origin, where we can melt the DNA and start replication. So we begin at these origins of replication. In eukaryotes, there are multiple places for that begin origination. In bacteria, it's one, and in bacteria, it looks like a bubble. As you separate the strands, you can see a replication bubble, if you will, where you see two double strands being created. So the double helix is open, and we have these special proteins that are designed to go in there like a wedge and open those two single strands apart, creating two replication forks, and we're going to synthesize away this way and synthesize away this way. So eukaryotic chromosomes have lots of them. Bacteria generally only have one, maybe two. Okay, but in either situation, we're synthesizing away, and then all these replications will merge together once you've completed the DNA replication. Very important point. Very, very important point. No matter what direction we head, we can only synthesize a new strand 5 to 3. We can only synthesize a new strand 5' to 3'. That means that we have to read the DNA 3 to 5 because remember it's anti-parallel. 5 to 3 and then 5 to 3. It's anti-parallel. So when we're synthesizing the DNA, like say, let's look over here. We would synthesize 3 to 5 in this direction, but we can't synthesize 3 to 5 in this direction because we would be reading the wrong way. Okay, so we reading 3 to 5, but if we were going this way, we'd be reading 5 to 3, and that's a problem. So we're going to show you how we overcome that. Here's an example of a bacterial chromosome that's being replicated. You see this beautiful replication bubble here. In bacteria, we know of one specific replication of origin. And so that's where those initiator proteins come in, create two replication forks moving in opposite directions. So this one's going to come around, this one's going to come around, and eventually they will meet. This in eukaryotic as well, but it happens in multiple places. So here's our original chromosome here with multiple places. So we see multiple replication bubbles in the same DNA molecule, and they merge together to form two semi-conservative daughter DNA molecules. So at each end of those replication bubbles, we have what is known as a replication fork. And in terms of their proteins and everything, they're identical other than their orientation of direction. So let's now get into the anatomy of DNA replication. By the anatomy, I mean the proteins and the strands and things along. And so we're going to be doing a little bit of molecular anatomy here. Our first bone, if you will, but this is a protein, is called a helicase. So after these replication forks have been created, we have to continue moving those replication forks away from the point of origin. And we are melting the DNA, and we've got this magenta circle around this one right here, this triangle, using an enzyme called a helicase. It is separating the two strands. It's working like a wedge, and it's breaking those hydrogen bonds, moving the replication fork. And we've got one. This would be right here on this one, and we've got one not shown on this one here, going that way. Now, as that helicase separates the strands, the DNA is going to want to rewind back into double strand again, or want to re-anneal is the phrase. So we have special proteins that will bind the DNA for just a split second, just a teeny tiny fraction of a second. They are called SSBs, single-stranded binding proteins, single-strand binding proteins. There's a set on the leading strand, and there's a set on the lagging strand. Okay. You should be able to recognize leading and lagging strand, and I'll work with you on that when we get a chance. It's hard to see here, but in any case, as that helicase separates the DNA, these little SSBs, single-strand binding proteins, are stabilizing and preventing the DNA from re-annealing behind the helicase. Now, as that helicase is going this way, because DNA is so coiled and so forth, we would create a real problem with twisting and binding. So we have this special third protein, and I guess it's got a little bit of a longer name, called a topoisomerase. And what it does is it will relieve the torsion on the unwound on the DNA that has yet to be unwound by nicking it for just a second and allowing it to unspin. So it'll nick one of the phosphate backbones, allow the torsion to unwind, and then re-anneal it real quick. So we have the helicase, the SSBs, the single-strand binding proteins, and topoisomerase. These guys are all leading the charge, moving that replication fork. Here's our replication fork here, one of the two. Here is our topoisomerase, and you know it's a topoisomerase. Why? It's beyond the replication fork. It's not melting the DNA, and it's also nicked one of the phosphate backbones, and that allows that single bond in the other phosphate to swivel around, so it'll relieve torsion. So there's our topoisomerase. What about this protein right here? It's moving into the replication fork. It's causing them to split. Well, that's helicase. Oh, what are these guys? They're binding one strand of DNA, single strand binding proteins. Well, what about this guy over here? Well, I've said we can only synthesize 5 to 3. We can't synthesize 3 to 5. The other issue that we can't do is that we can't just synthesize DNA. We have to have a 3' end to synthesize onto. So now we're going to add the next protein to this mix, and this is called primase. This is an RNA synthesizing protein. So this red right here is not DNA, it's RNA. It synthesizes a few nucleotides of RNA, a little primer, and then the DNA polymerase can come in and finish the job synthesizing in on the leading strand. So that's a little RNA primer. It synthesizes a little bit of RNA. It binds to that strand. We're going to do this over here, but we're going to do it in a slightly different way. I'll show you in a second. So we're synthesizing a little RNA primer. And then so here's the primase. Synthesizes a little RNA primer. Again, synthesizing 5 to 3, reading 3 to 5. Okay, so we have that RNA primer. Because it cannot initiate synthesis, a DNA polymerase cannot initiate synthesis of a polynucleotide. They can only add onto a 3' end, so they create a little primer there on which to begin synthesizing the other strand of DNA. So there's that primer. Once the primer is maybe 8 or 10 nucleotides, the DNA polymerase comes in, binds, and then starts synthesizing DNA. Again, 5' to 3', reading 3' to 5'. So a primase can start an RNA chain from scratch. It doesn't need a 3' end. Okay? So we're going to synthesize a little bit of primer, and then DNA polymerase can come in and start synthesizing DNA. It's only 5 to 10 nucleotides, very, very quick. So these polymerases catalyze the elongation of DNA at the replication fork. And they're doing it on both strands, but because we can only synthesize 5 to 3, it creates a little bit of a technical problem on that lagging strand. There are 2 different kinds of DNA polymerases, and this can be a little bit confusing, so a little confusing here. Our main DNA polymerase, our primary DNA polymerase, is called DNA polymerase 3, Roman numeral 3. DNA polymerase 1 we're going to use on the lagging strand, more so than the leading strand, a lot more so than the leading strand. They have a very, very important difference, and that's why they have different functions. In the case of bacteria, they may get 500 nucleotides per second, and about 1/10 of that in human cells. That's because bacteria generally aren't as concerned about errors as they are in eukaryotic cells. Part of their genetic diversity occurs because of mutations occurring during DNA replication in bacteria. So we're adding nucleotides, so nucleoside triphosphate, all right, deoxy ATP. Regular ATP, by the way, is a ribose nucleotide. This is deoxyribose ATP, deoxyribose GTP, and so forth. So it's similar to ATP, and it actually supplies the energy. These molecules supply the energy for elongation. As we hydrolyze the phosphates off of here, that provides the energy for DNA elongation as we're replicating it. So deoxy ATP, regular ATP, and the other ones that don't have that little lowercase d are ribose nucleotides. Okay. So it joins the DNA strand, it loses those 2 phosphate groups instead of 1. Like for instance, when we make ATP and turn it into ADP, releasing energy. This one, we're actually releasing 2 phosphates. And so instead of P subscript I, inorganic phosphate, we have 2 phosphates linked together, and that's why it's called pyrophosphate. So here we are adding a nucleotide to that 3' end, and here's that nucleoside. So nucleoside is the base and the sugar, triphosphate. So we're adding this triphosphate nucleoside. DNA polymerase removes those 2 end phosphates and creates a bond here between the 3' OH and that first phosphate. So now we have added that letter T here. This is T. Here's T over here. We've created that phosphodiester bond there. Because it's anti-parallel, we have to overcome the issue of synthesizing 5 to 3 because we can't read 5 to 3 and synthesize 5 to 3. We can only read 3 to 5, synthesize 5 to 3. So it's DNA polymerase can only add to the 3' strand. It can only elongate in the 5 to 3' direction. So this is just fine here. But what about the other strand? The other strand works like this. And I'm just going to show you this briefly. As enough DNA becomes available, we'll synthesize a little primer, and then we'll synthesize DNA until we hit to that previous primer. As more DNA opens up, a primase will get in here, make a little primer, synthesize DNA until we hit this primer. These fragments appear, alternating DNA with RNA. Those fragments are collectively known as Okazaki fragments for the researcher who first discovered them. And so we'll show you this more and more. Okazaki fragments are the fragments of DNA that are made on the lagging strand. You notice that we only have 1 primer on the leading strand. That's because we can synthesize continuously as that replication fork moves down the DNA. You don't have to add another primer. But what's happening here is we have to create some space. Primase comes in, makes a little primer. DNA polymerase 3 synthesizes DNA until it hits that previous primer and then stops. So this Pol 3 here, polymerase 3, is almost done. It's going to get right up to that primer, synthesize that last nucleotide, and then fall off. So here we can see primase create the primer, and then polymerase synthesizing after that until we hit the previous primer. And there's our Okazaki fragment. Over here on the bottom, this is the leading strand. That's the DNA polymerase synthesizing right into the replication fork. And then we have the primase come in on the lagging strand. The primase will bind here, and then we'll get that DNA polymerase 3, and we will repeat the process this way. This whole process is known as semi-discontinuous. We are semi-conservative in terms of a parental strand and a daughter strand. But in the DNA replication, we are semi-discontinuous. We are continuous here. This is continuous. This is discontinuous because it keeps stopping. And so it is semi-discontinuous. Since we're right at the half-hour mark, I'm going to go ahead and stop here. Again, we've created the leading strands using helicase and single-strand binding proteins and topoisomerase, not shown in this picture. We get the primase, make a little primer, and we synthesize DNA with DNA polymerase 3. Only on the lagging strand, we're going to do this in a discontinuous fashion. As space opens up, we prime a little bit, synthesize DNA from the previous primer. So we created this one first, we created this one second, we created this one third. So we're going to stop right there, and I will pick up with the final 20 slides or so, finishing off this mechanism. Take this slow, work methodically, and you'll get it. It's not as hard as it seems. It's a lot of easy things all working together simultaneously. And I'll talk to you soon.